Cells are the smallest structural and functional units of an organism. These cells provide structure for the body, taking nutrients from food and convert those nutrients into energy. And also, cells reproduce to replace the dead cells or for growth of that organism. Based on number of cells present in an organism, organisms are classified into unicellular and multicellular organisms. Organisms like bacteria, paramecium, amoeba, and archaea are made up of only single cell, and the whole life activities are carried out in that single cell. These kind of organisms are called unicellular organisms. If we closely observe these organisms, we can notice that bacteria and archaea does not contain a nucleus, while paramecium and amoeba do contain a nucleus. So, unicellular organisms are further classified into prokaryotes and eukaryotes. Prokaryotes are always unicellular, while eukaryotes can be unicellular or multicellular. For billions of years, these unicellular organisms are gradually evolved into much more complex multicellular organisms. Organisms like animals, humans, plants are made up of more than one cell and are called as multicellular organisms. Unlike an unicellular organism, where all life activities are carried out in a single cell, in a multicellular organism, similar cells combine together and forms a tissue, and different kinds of tissues work together and forms an organ, and different kinds of organs work together and forms an organism. And next is history of cell. Usually cells are microscopic, and invention of a microscope during 1600s led to the discovery of cell. And Zacharias Janssen was the first known person to build a microscope, and this is how the microscope looks like. After that, in 1665, an English scientist called Robert Hooke improved the already existing compound microscope and achieved a better magnification. And this is how the microscope looks like. When he placed thin slices of cork under his microscope, he observed that the cork is made up of small room-like structures like in a honeycomb. He called them as cells. However, Hooke observed were dead cells of the cork, and the first known person to observe live cells was Antony van Leeuwenhoek. Leeuwenhoek built a single lens microscope which looks like this, and it can magnify objects up to 300 times. He observed several unicellular organisms from a pond water. These are some of his observations, and he called them as animalcules. Later, these animalcules are renamed as bacteria. Along with pond water, he also observed our bodily fluids, and these are some of his observations in a bodily fluids. Because of his observations on microorganisms, he is acknowledged as father of microbiology. And the next breakthrough came in 1831. Scientist called Robert Brown observed a nearly circular spot in epidermis of orchid leaf cells. It is slightly non-transparent, and also he noticed similar structures in other cells as well. He came to a confirmation that the structures were internal parts of the cells, and he called them nucleus. After that, in 1838, two scientists, Sleeden and Schwann, combined together proposed the cell theory. Cell theory composed of three parts. Number 1. All organisms are made up of one or more cells. Second one, cell is the basic structural and functional unit of an organism. However, these two scientists doesn't have any idea about cell origin or cell division. That's where another scientist called Rudolf Virchow stepped in with his observations, and he proposed the third one of the cell theory, that all the cells come from pre-existing cells. This is the quite interesting history of cell, and next we are going to learn about structure and functions of a typical animal cell. A typical animal cell is composed of various parts, and let us start with the cell membrane. Cell membrane is the outermost layer of the cell, and it is also called as plasma membrane. Cell membrane is semi-permeable, which means it only allows selective substances to pass in or pass out. Cell membrane defines the size and shape of the cell and encloses the cytoplasm. The jelly-like fluid which is filled inside the cell is called cytoplasm. Cytoplasm is composed of salt, water and enzymes, and helps in transportation of different kinds of materials which are required for cells' daily activities. And also, cytoplasm is the portion where different kinds of organelles are suspended. So what is an organelle? Organelle are like little organs with their specific functions. Do you know what are their functions? Now we are going to learn about functions of different kinds of organelle in an animal cell. And let us start with the largest organelle, nucleus. Nucleus is the control center of the cell because it regulates and controls the activities like growth and metabolism. Nucleus is the largest organelle, occupying more than 10% of the cell volume. Nucleus also contains nucleolus, surrounded by a tangled form of genetic material called chromatin. During cell division, these chromatin condenses into chromosome, and nucleus produces the ribosomes, and through nucleus pores, these ribosomes are then released into cytoplasm. Ribosome. Ribosomes are made up of two subunits, a large subunit and a small subunit. Ribosome synthesizes the proteins, so ribosomes are also called as protein factories. Ribosomes released from the nucleus may wander freely in cytoplasm or may attach to endoplasmic reticulum. Endoplasmic reticulum is a continuous membrane system, lies adjacent to nucleus, and it forms a series of flattened sacs. Although endoplasmic reticulum is made up of same membrane, it is divided into rough ER and smooth ER. Surface of the rough ER looks rough because of attachment of ribosomes, while smooth ER looks smooth. Smooth ER manufactures the fat molecules or lipids and packages them into vesicles and sends them to places they need. Rough endoplasmic reticulum packages the proteins that are synthesized by ribosomes into vesicles. Vesicle means a small sac, and these vesicles are then transported to Golgi apparatus. Golgi body. The vesicle that is pinched off from rough ER reaches the Golgi body, and the Golgi body further modifies by folding them and by adding the materials like lipids and carbohydrates, and once again made into vesicles and pinched off to various parts of the cell. Golgi body packages the waste material into vesicle for extraction purpose, and also packages the proteins into a membrane-bound organelle called lysosome. Lysosome. Lysosomes are membrane-bound organelles which contain digestive or destructive enzymes. Lysosomes digest the excessive or worn out parts of the cell. Sometimes degrade the foreign bodies like bacteria. In some situations, like when the cell is damaged, these lysosomes burst and digest its own cell, resulting death of that cell. So, lysosomes are also called as suicidal bags of the cell. And next organelle is centrosome. Near the nucleus, we can find the centrosome, and as shown in figure, centrosome is made up of two centrioles perpendicular to each other. During cell division, chromatin condenses into chromosomes and centrosome duplicates and moves apart to the poles and produce microtubules. And these microtubules pull a copy of each chromosome to opposite side of the cell, which results in equal distribution of DNA, and two identical cells are formed. At last, we are going to learn about an organelle that produces the energy that is required for the cell activities. Mitochondria. Mitochondria are small spherical or cylindrical organelle, and size of the mitochondrion ranges from 2 to 8 microns long and about 5 micron wide, and it is 150 times smaller than nucleus. Mitochondria are double membrane organelle which contains an outer membrane and inner membrane. An inner membrane contains folded like structures called cristae, and the space between cristae is filled with fluid called matrix. Mitochondria combines the sugar with oxygen to produce ATP. ATP provides the energy that is required for the cell activities. So mitochondria are also called as powerhouse of the cell, and combining down sugar with oxygen is called cellular respiration.